Hello everybody and welcome. In this video, the third year class of the Quadriennale course will take you back through the history of the discovery of DNA, to then let you enter the laboratory to roll up your sleeves to carry out a DNA extraction together. Let's start by talking about the father of genetics, Gregor Mendel. Although he never talked about genes or DNA, he was the first to discover the principle of character inheritance. His studies laid the foundations for following researches by scientists such as Miescher, who, studying white blood cells, discovered what he at the time called nuclein, and which we know today as DNA. At this point, a scientist named Sutton wondered where this nuclein came from, and assumed that it was contained in chromosomes. Morgan, instead, observing the flies with white eyes instead of the usual red ones, wondered if the genes could mutate, but it was Mueller who found an answer to it by discovering that X-rays cause mutations in chromosomes. In the scientific community, a debate arose as to what this mysterious transforming factor was. Many biologists believed it depended on proteins, others on DNA. To find out, the scientists Alfred Hershey and Martha Chase conducted an experiment based on the property of bacteriophages to replicate inside a host cell. They labeled the proteins of a certain number of bacteriophages with radioactive sulfur 35. And the DNA of the same number of other bacteriophages with radioactive phosphorus 32. At that point, the bacteriophage was allowed to infect the bacteria, and when the infection was over, they used a blender to detach the bacteriophages from the bacterium cells. Then, they checked the presence of radioactive cells, and it was found in the samples with phosphorus, determining that the DNA was the transforming factor. Finally, we come to Watson and Crick, the biologists who discovered the structure of DNA. Thanks to the important contribution of the chemist Rosalind Franklin, who took the first photo of a DNA strand in history, the two scientists identified DNA as two intertwined strands made of nucleotides consisting of three components: a phosphate group, a sugar group, and a nitrogen base that can be adenine, guanine, cytosine, or thymine.
Now, let's talk about DNA and RNA structures. First of all, DNA plays a key role in the protein synthesis process. In fact, in its structure, there are protein coding regions called exons that are not continuous and are interrupted by non-protein coding regions called introns. RNA is created starting from DNA in the process of protein synthesis. Both DNA and RNA are made up of simple repeating units called nucleotides, which consist of three component parts: a sugar group and a phosphate group, which combined form the backbone of the structure, and a base. DNA's sugar is different from RNA's one. Deoxyribose sugar is found in DNA and it differs from ribose sugar, found in RNA, by the lack of a single oxygen atom. Both DNA and RNA have four possible bases. Adenine, guanine, and cytosine can be used in both the nucleic acids, but uracil is found only in RNA, replacing thymine, that is only present in DNA. If we have a look at the structures, we can see that if RNA has only one strand, DNA on the other side consists of two intertwined strands that hold together thanks to the hydrogen bonds between the bases. In DNA, adenine always pairs with thymine and cytosine always pairs with guanine. In these pictures, we can see giant lamp brush chromosomes. These are found in amphibian eggs. They have lateral loops of DNA that produce a brush-like appearance. The last thing we are going to talk about is an experiment that has the aim to separate the DNA that is located inside an eukaryotic cell and analyze its structure. The materials needed to carry out the experiment are half banana, distilled water, washing up liquid, ethyl alcohol, pineapple juice, salt. And this is the equipment required. The first step is to chop and smash the banana in a bowl using a fork. Separately, in a beaker filled with distilled water, add a spoon of washing up liquid, a pinch of salt, and stir it gently, avoiding making bubbles and foam. Add 9 centiliters of the liquid to the banana and mix it slowly, still being careful. Now the salt added before is eliminating the proteins, and the washing up liquid is destroying the lipid membrane of the cells and the nucleus. When a homogeneous mixture is reached, use a colander to let the liquid drain through. Then put 5 centiliters of that concentrated DNA into a test tube and add 3 centiliters of pineapple juice to it. Mix gently because that helps to demolish proteins. Finally, the last step is to add 6 centiliters of ethyl alcohol in the test tube and wait for the result. Since DNA is alcohol insoluble, it appears as white strands between the solution and the alcohol. What's interesting about this experiment is that even though the steps are quite simple, you still have to be very precise, because if you don't, the result can look very different.